My name is Beth Cimini. I'm the lead image assay developer in the imaging platform, which is also known as Ann Carpenter's group at the Broad Institute. Probably many of you are more familiar with our software CellProfiler, which does image analysis and quantification. But it has a companion program called CellProfiler-Analyst, which is for data exploration and machine learning. So we think of these as two separate halves that are designed to work together, but certainly don't have to be used together. CellProfiler is what allows you to take your pictures and turn them into spreadsheets or databases of measurements. CellProfiler-Analyst is how you use those to answer a question. If you want to learn more about CellProfiler-Analyst, you can check out the previous stream that we did 2 weeks ago, where I give a basic overview as to how that software works. So why is it important to create a tool like CellProfiler-Analyst, and what value does it add to you doing your science? This is a study that our lab did a few years ago, and I think that this is changing a little in the field of biology, but really not all that much. When you think about high content screens, or you think about groups doing large batches of image analysis, for the most part, people only report 1 or 2 aspects of their cells or their organism or whatever it is that they're measuring in their images. The vast majority only look at 1 to 2 features. So this could be something like cell area or intensity of a certain protein in the nucleus, and a few of them report 3 to 5 or even more than 6, but really it's not much. People who use CellProfiler tend to report more features a little bit more, but it's still not very common. Now you might be thinking, well, if I know the thing that I care about in my cells, I know specifically how my cells are different before and after the treatment that I care about. Who cares? I should only report the 1 to 2 features that I know are the important thing. And a lot of good biology really does happen this way. This is a collaboration we did with John Crispino's group at Northwestern several years ago, looking for drugs that cause cells to stop dividing, specifically this particular type of leukemia cell. And certain drugs did, as it turns out, quantifiably show that the amount of integrated DNA content in the nucleus went way up, indicating those cells were continuing to duplicate their DNA but no longer dividing, and allowed us to show drugs that are now in clinical trials. So sometimes you might only need 1 measurement to find the phenotype that you care about. And if you only need 1, that's great. Sometimes you might need 2. You can usually get cell cycle phase, for example, with 2 measurements. If you have just a measurement of DNA content and a measurement of mitosis, you can get whether a cell is in G1, S, G2, or which phase of metaphase, or of mitosis, metaphase, prophase, anaphase, or telophase. And again, if you only need 2 measurements to find the thing that you care about, then that's fine. The problem is there's lots of weird, wonderful biology out there in the world, and some of it isn't as easy to break into 1 or 2 measurements alone. There probably are measurements that would help me look at all of these different cell phenotypes, but I certainly don't know the names of them off the top of my head. I couldn't tell you which measurements are the ones that are going to distinguish all these different classes of cells. And frankly, there's no reason for me to have to do that. As long as I, the biologist, can tell the difference between them and know what the classes are, in the same way that with CellProfiler we talked about the computer doing the part that it's good at in doing quantitative measurements, here with the data analysis, we can let the computer do the part that it's good at, which is now taking this giant summary of measurements and figuring out which are the ones that are actually important and which are the ones that find the objects we care about and the classes that we care about. So we think about this, we sometimes refer to cells having a morphological profile or a cell morphology fingerprint. So rather than having 1 or 2 measurements that we know describe the phenotype that we care about, we can just as easily work with dozens or hundreds or thousands of measurements for every cell. And then when we look across many cells, we can see that some cells will fall into different groups or different categories. And those might be interesting phenotypes that we care about. So by taking lots of measurements, rather than just one or two ahead of time, we can actually find lots of categories of cells, possibly even some categories or phenotypes we didn't even know going in might be present because we have enough measurements to see lots of different groups. So that's why we have both CellProfiler and CellProfiler-Analyst, because we want you to find the objects that you care about in your image, but we want you to then make lots of measurements of them so that you can then potentially have measurements that allow you to find really interesting phenotypes later. This comes back to our lab motto, measure everything and ask questions later. It's also the name of our blog. So in our lab, we typically go through a workflow where we try to build up big measurement suites to find lots of measurements of cells, even ones where you think you know what the phenotype of interest might be. And as before, I want to emphasize that many of these metrics can be found in lots of tools. This isn't something you can do only in CellProfiler. And then we go into CellProfiler-Analyst, which allows us to take the measurements that came from CellProfiler and explore them in lots of ways. You can just look at the columns from the data, you can create scatter plots, you can look at a whole plate of data, and you can do classification. And so we're going to go through all of that together today. But the part that I think will be most new to people and most interesting, that where CellProfiler-Analyst makes something that is technically quite challenging very easy, is the classifier tool and the ability to do machine learning. So if we've generated enough measurements ahead of time of our cells, we can imagine that all of these measurements are associated with every cell in CellProfiler-Analyst as these little gray lines that you can see here next to each cell's picture. Now you, the biologist, know what the phenotype is of the cell that you care about. And so in this toy example, we can pretend that the biologist cares about these sort of yeast-looking cells. So the biologist can say, is this a phenotype that I care about or not? Is this the phenotype that I care about or not? And so on and so forth. And as long as we have measurements associated with these cells, the computer, which is good at looking at lists of measurements and figuring out if some of them apply more to some cells than others, can go through and decide that, for example, certain measurements do a good job of describing only the cells that we care about and not the cells that we don't. We call this doing machine learning or machine learning-based classification. Now, the wonderful part here is you don't need to know what the rules are or even ever see what the rules are. As long as we iterate through this and it keeps showing you cells and you agree with how it's now classifying them into the cells that I care about or not, the computer can handle rule selection and you just focus on your biology, which is of course the part that you know best. Just to show that this isn't just in the basic science lab, but this also can be used in the clinic, this isn't a paper that used CellProfiler-Analyst, but it is a paper that used cell profiler features and this same machine learning type aspect to do drug selection for patients. They had patients who had recurrent blood cancers, and they took the blood cells from these patients, they put them in an automated microscope plate and treated them with lots of different clinically approved diseases. And after taking lots of measurements of the cells, they created a machine learning based metric of whether or not cells were alive or dead or dying. They could then see whether the cells that were dying were the patient's normal cells or the patient's cancer cells based on a specific marker for each patient. So using this machine learning classifier, they can now tell the difference between a drug that's killing all cells, a drug that's killing cancer cells only, a drug that's killing normal cells only, or a drug that isn't killing anything. And in cases where the doctors had a good idea of what drug the patients might want, the physician chose the treatment, and that was the case for 13 patients. But in the cases where there really weren't other drugs that were obvious to the clinicians which drug to try, they allowed this machine learning guided algorithm to decide which drug the patient should receive. So this is how the patients had done on their previous round of chemotherapy. Again, all of the patients in this had had at least one recurrence of cancer before, which is why this goes down to 0, because all of the patients had relapsed before. And you can see they tended to relapse pretty quickly, within about 3 months for almost everyone. But for the patients who got drugs that were aided by machine learning, you can see that those patients tended to do much better than they had on whatever their last treatment was. Took many of them much longer to relapse, and some of them did not relapse at all within the time period of the study. And looking at drugs that were chosen by the physicians in gray versus drugs that were chosen by machine learning in blue, you can see that there are gray and blue dots in both cases, and there are people who received physician-guided drugs or machine learning-guided drugs who did both sadly pass away, but there's definitely improved disease state in patients who got machine learning-derived drug recommendations. So this isn't something that's just an academic exercise. This is something that can actually go into the clinic and change lives. So this sort of machine learning analysis is actually extremely powerful. We're not going to be curing anybody's diseases today, but what we are going to be working with is based on real data and is based on finding a drug that causes a particular phenotype. This is real cell data where cells were treated with a dose of wortmannin that causes a certain transcription factor tagged with GFP to go from the cytoplasm into the nucleus. But the dose that we're using here as our positive control dose is actually a pretty toxic dose. So what the researchers wanted to determine was, can we find a lower dose that does this well? Can we calculate an IC50 curve for this drug? And that's exactly what we're all going to do together right now. The data and the exercise we're going to go through today are all based on one of our tutorials, and you can find these tutorials online in our GitHub repository. Specifically, what we're going to be going through is what we call our translocation tutorial. And you can see here it has data and it has a PDF of instructions. So if you want to go through all of this exercise at home by yourself later, you have the instructions to do that. I have this same folder here with all of the stuff that I mentioned to you, and I have my copy of CellProfiler open. We're just going to use CellProfiler very quickly to generate our measurements, and then we're going to use CellProfiler-Analyst for everything else. You see here I have a start and a final CellProfiler pipeline, or CP pipe file. If you're going through this exercise by yourself from the beginning, I definitely recommend you open the PDF, you go with the start pipeline, and then you go ahead and build the pipeline yourself. But for the purposes of today's exercise, we're just going to use the final pipeline. Ask if I want to import it, and I tell it yes. And I have a folder here full of images. I'm going to drag and drop here into the images module. If I double click on these, you can see the images come up. And I have my nuclear stain, and I have my GFP stain. Because this pipeline's already built, I don't have to do a lot, but I do want to actually add some metadata into this pipeline. So I told you that this was a drug screen where they were adding different doses of drug. So there's a CSV included in this also that I've opened up here in Excel. And what you can see here is that we have a list of by well, what dose of drug was each cell treated with. And is this a positive control, which is marked as a 1, a negative control, which is marked as a 0, or one of our experimental drug groups. So it's marked as 0.5. Now, I don't need to add that information into my image analysis pipeline. My image analysis pipeline will run the same with or without it. But if I add it now, then CellProfiler-Analyst has access to this information after the fact. It also means that if I need to go back to this experiment in days or weeks or months later, I don't have to go through and dig up the information of, wait, what dose was each of these things treated with? I just already know, because that information is in my image analysis files along with the rest of my data. So I have a CellProfiler pipeline that's already set up to pull some information from each image's name, specifically what well, what site within the well, what channel, and what plate it came from. And the fact that for each image I can tell what well it came from by the file's name means that I can use the fact that I know for each well what dose it was treated with, that I know now for each image what dose it was treated with. And I do this simply by loading in the CSV. And by saying that the piece of information called well that it pulled from the image's name is the same as the piece of information called well that it found in the file. So when I hit this update button now, you can see that for each image, I now have a column that's called dose. And so for each image, it's now already figured out what dose of drug that image represents. If you want more information about this, or other kinds of information you can associate with your CellProfiler pipelines, the metadata module's help button, which is right here, will tell you a lot more information. You can also find more information, for example, about how to set up one of these extractions so that you can extract important information from your own files' names. But for now, we're going to move on, since this is a CellProfiler-Analyst tutorial. I've made, on my desktop, an output folder that I want to send my output to, and I'm going to go ahead and just hit Analyze. I'm running 8 workers, and I have 26 image sets, so this should be finished within a minute or two. When I open up this PDF, again, the first part of this is going to be the instructions for creating that CellProfiler pipeline. And again, I definitely recommend, if you want more practice with how to configure a CellProfiler pipeline, you do this on your own. But if you just want to practice how to use CellProfiler-Analyst, you can scroll down to where it says Using CellProfiler-Analyst software about 2/3 of the way through this PDF, and that will show you how to actually use this software if you prefer to read things as opposed to listen, as I'm doing right now. I'm just going to give this a minute. So far what's in it is a database. So we're using the Export to Database module here because that's what CellProfiler-Analyst needs. You can also export the information into a spreadsheet. It's really whatever you're most comfortable working with. I will say, however, that even if you're a little overwhelmed or intimidated by databases, if you use the SQLite type that we've mentioned here and that we're using here, it's really not that much different than actually just using a spreadsheet. An SQLite is essentially just a wrapper around a few different spreadsheets that are all living in the same file. And in just a minute, when this is finished running, I'll open that up and I'll show you exactly what that looks like underneath. The Export to Database module also has an option to create what we call a properties file, and that is a file that tells CellProfiler-Analyst how to look at your images and how to figure out what constitutes a plate, what constitutes a well, what size of images should it show you, what color of images should it show you. All of that information is configured by your properties file. And the properties file is just a text file, and I'll show that to you as well. So if you've analyzed data in the past and you actually want to use CellProfiler-Analyst on it now, you don't actually need to go through and reanalyze the data just to make one of these property files. It's just a text file. So you can run, for example, an example pipeline, make an example properties file, and then adapt it to your own data. It's pretty simple. Okay, that didn't take too long. So I have my output images now that I made, showing the cells that were identified in my pipeline. And you can see as I scroll through them that some of them the GFP is nuclear, and some of them the GFP is cytoplasmic. And at the end I have these two files I mentioned, my database and my properties file. When I open up my database using a program like DB Browser or any other database program that you like, if I go to Browse Data and I look at, for example, my image table, you can see that ultimately this database looks pretty similar to just what you might otherwise see in an Excel spreadsheet. We have columns that all have particular kinds of measurements in them, and then each row in this image spreadsheet represents an image. In fact, if I want, I can even export some of the information in this to a CSV or import information that's in a CSV into a database. So there's nothing to be scared of with SQLite databases. They're actually really simple and easy to use. If your university or company has access to a MySQL database, those are a little more complicated, but they're great for example, if you're running big data sets on a cluster, because then many copies of CellProfiler can all write to the same output file simultaneously. But if you don't have access to one of those, there's no problem. You can run this just fine on your local machine the way I just did. This properties file that I mentioned, and you can see when I click on it, even though it has a little bit of a strange extension, it is just a text file. And what it says is that my information is in an SQLite database, that SQLite file is on my desktop in a folder called translocation output, that when it wants to know what well images are in, it should go to image metadata well, that I have two channels, DNA and GFP, and that it should show those in red and green. Now some of you might be red-green colorblind, so let's just go ahead and edit this and say that we want to use magenta and green. I also could have done that inside CellProfiler, but just to show you, it's really easy to adapt this properties file so that you can do whatever customized viewing in CellProfiler-Analyst that you want. There's a lot more information about, for example, what plate type or how big of an image it should show me when CellProfiler-Analyst opens, but a lot of that's going to be more understandable if I just go ahead and open CellProfiler-Analyst. So when I click on the CellProfiler-Analyst icon, it brings up this menu saying that it wants me to select the file that contains my properties file. So here it is. And if I go ahead and hit open, I get a menu that looks like this. And in my CellProfiler-Analyst menu, along the top, it shows all the different kinds of tools I can use. The simplest one is just the table viewer. I can use it to just load any table from the database, and if I didn't have a program like DB Browser that I wanted to use to look at the per image table, I can do it here in CellProfiler-Analyst, and I have all this access to the same information and the same data within this program. I can open the image gallery, and I can say that I want it to show me the first few images. I can also open the plate viewer, and here, if you have data that's in a multiple plate format, you get a really nice overview of any parameter that you want from your experiment. So, for example, if I want to look at the dose information, I can do that here. And you can see the way this plate was laid out was their negative control wells were in the top half of column 1 and the bottom half of column 12. The positive control wells vice versa, the bottom of column 1 and the top of column 12. And across the middle here was my dose response curve. If I change the slider here so that we can see more, it becomes a little more obvious that that's where my dose response curve was. It's not that easy to see just because this was a log dose curve. I can look at any of the other measurements, not just the ones I took per image, but I can look at a per well average of any of the measurements I took per cell. So for example, if I want to look at the nuclear intensity of the DAPI channel, well, that isn't a measurement that I took. But if I want to look at the nuclear intensity in the GFP channel, say the mean intensity in the GFP channel, that is a measurement that I took. So I can go ahead and I can graph that. If I had wanted different measurements, I could of course always go back to my CellProfiler pipeline, go to the Measure Object Intensity module, and add more measurements. But I think what we have now will do. And so if I look at the amount of GFP that's in the nucleus, these are my negative control wells and these are my positive control wells, and you can see there's more GFP in the nucleus in my positive control wells, which makes sense. If we didn't see that, we'd be a little bit worried. I can go beyond just looking at the data here. I can also look at a thumbnail of the image. So if I want to look at my GFP image, I can just double click in that well, and it pulls up my GFP and my DAPI. If I look at from the positive control, there you can see it. I can adjust things in this window as well if I want. I can play with the brightness contrast. I can go from linear mode to log mode. I can even adjust my zoom. So the plate viewer tool can actually be a really valuable way to, when you first finish an experiment, to get a sort of high-level overview of what's going on. You can say, I think I know what are the most important measurements. Let me scroll through and find which ones I think I care about. If I look at the intensity in the cytoplasm, then we see the opposite of the relationship we saw before, that most of the GFP intensity is in the cytoplasm in the negative control wells, which is what we thought before. Can also do histograms, density plots, and box plots, which I won't go through, but this is the same as with any piece of statistical software that you might use. What's special and unique to CellProfiler-Analyst is this classifier tool. When I open this window up, it's going to show me two bins. Because to do a classification, you need at least two different kinds of phenotypes. I'm going to change the names of these, though you don't have to. You can just leave them as positive and negative. I'm going to call this one nuclear GFP, and I'm going to call this one cytoplasmic GFP. Now, I'm going to ask CellProfiler-Analyst to find me 20 random cells. You see I get a mixture of cells. I'm going to increase the zoom here just so I can make sure that everyone can see this at home. I'm also going to increase the brightness a little bit. So here are the cells. And all I need to do now is, based on my knowledge of the biology, is drag and drop them into these bins. If I hover over any one of these tiles, it shows me which cell is actually the cell, if there are multiple cells, by putting a little box over the center. So I'm going to start by just grabbing a few. And you can see I haven't actually even looked at the measurements yet. I'm just sorting the cells by the phenotype that I see. We offer a few different classification algorithms, and it's okay if you don't know the difference between them. This information is all on the CellProfiler-Analyst website if you have more questions. I'm going to pick fast gentle boosting here because that's a method that works really nicely, but you can pick whichever one you like. If I go ahead and I hit the train button, CellProfiler-Analyst is now going to take a look at all of the measurements that were present in that whole list of measurements that we saw here in the plate viewer tool. When we looked in our per object, we saw that we have many, many measurements, and it's going to show me what measurements it thinks does the best job of separating these 5 cells from these 5 cells. And the ones that it picked are all metrics of correlation in the cytoplasm between the GFP and the DNA. This makes sense since it's a translocation assay. What we want to see is essentially if the GFP is highly correlated with anything else in the cytoplasm or not. I can hit the evaluate button, and I can see how good a job the rules that it picked are at actually separating these 5 cells from these 5 cells. And it turns out that so far they're not great. It's 75% accurate at figuring out if cells that are actually in the left bin should be in the left bin, and 60% accurate if cells that are in the right bin should be in the right bin. It's not great, but we've only just gotten started. So let's fetch 40 more cells. And I want to get up to 10 or 15. And the cells that I have, you can see they have a range of brightnesses in terms of their actual green brightness. They have a range of shapes, they have a range of sizes, and all of that is good. You want your training, what we call our training set, which are the cells that are in these bottom bins, to be relatively representative of the diversity that's present in the experiment. The closer these are to representing all of the different kinds of phenotypes that are cytoplasmic GFP, the more likely the algorithm is going to learn something and actually figure out how to correctly split these. If I hit train again, I can now try to evaluate again. You can see that we now have a slightly different list of measurements, although they're all still measurements of correlation in the cytoplasm. And these numbers have now gotten a lot better. It's 95% accurate at identifying which cells belong in this bin, and 88% accurate as the ones in this bin. That's pretty good. That's something I'm happy with. You might have noticed there are a few cells I haven't even tried to classify yet. Some of the cells in this experiment really don't have much GFP in them at all. I could just ignore them, but CellProfiler is going to try to give them a label, whether I classify them or not. So I'm going to go ahead and just make a class that's called no GFP. Because they're a pretty tiny fraction, it won't change the output of my experiment all that much, but it's nice for me to know how to split these, and I feel better having sorted all of the different kinds of cells, not just the initial 2 that I was looking for at first. Now that I've trained a bunch of different kinds of cells, I want to be able to save this. So if CellProfiler-Analyst crashes, or just if I want to stop working on this for today and come back to it later, that I have a record of what are the cells in this bin, these bins. That also might be important because if you want to see actually how the classifier was made later, it's good to know what actually were the individual cells that you said defined being a cytoplasmic GFP cell or a no GFP cell. I'm going to go ahead and save. Again, we call these things down here our training set. So I'm going to call this file just my training set. It goes here into my output folder. And it has here all of my list of cells that were in the nuclear GFP bin, the cytoplasmic GFP bin, and the no GFP bin, what image number they came from, what object number they came from, and all of their measurements. So I could go back and basically recreate later this sorting algorithm. I could also, if I were starting from scratch, load that training set. This is great because now I have 3 different bins that all have at least a few different cells in them, but I haven't trained yet for my 3rd class here. So let's go ahead and train. Now you can see we have a bunch of different kinds of measurements starting to come in. We have measurements about the maximum intensity in the cytoplasm. We also have ones that are called mass intensity ratio. And that's actually a measurement we made in CellProfiler by dividing the intensity of the GFP in the nucleus by the intensity in the GFP of the cytoplasm. We're able to calculate that inside CellProfiler, so that CellProfiler-Analyst has access to that, and it knows for any individual cell what its GFP intensity is compared to its nuclear intensity. And now that we have a no GFP class, it's saying that that has become a more important measurement in figuring out what's going on in our different classes. If I hit evaluate, you can see it still knows pretty well the difference between these 2 classes. It's not as sure yet about the no GFP class, but I didn't have many cells to put in there. If I want more cells, one thing I can do is I can ask it to give me a particular image. So image 1 was one of our negative control images. And when I ask it to show me what's going on in image 1, it shows me that there were 302 cells. One of them it thinks has nuclear GFP. 89 of them it thinks have cytoplasmic GFP, and 212 it thinks it has no GFP at all. I'm not sure that's right, so we're going to help correct CellProfiler-Analyst because it still hasn't quite learned what we want to do yet. Just try and make this image a little smaller so that we can get back to our bins. And if I go to show controls here, I can ask it to show me just what it thinks is cytoplasmic GFP versus no GFP. So let's start with what it thinks is cytoplasmic GFP. I generally agree with that, though it's definitely missing some where I can see some GFP in the cytoplasm. So now let's take a look at what it thinks is no GFP. Yeah, and there's some stuff there that I think is in the wrong bin, that it hasn't actually learned quite yet. So I can go ahead and actually drag and drop from this image into the bin over here, and I can use this to generate more examples of what my different classes are. Now I do need more no GFP cells in this bin. I don't want to take them all just from one image, because then it might think that the important thing for being no GFP is coming from this one image. So I don't want to give it too many examples, but I'll give it a few. I don't see any others right now that I really desperately want to correct. So I'll go ahead and close that one. Let's just try to score one more image. Let's score image 26. This is another negative control one. I'm going to score image 25 instead, just so I can get some examples of positive control. So for this one, it thinks that there are 256 cells, 182 of them have nuclear GFP, 55 it thinks have cytoplasmic GFP, and 19 no GFP. I'm a little surprised by how high this number is, so let's go ahead and take a look at what's going on there and see if there are any that we need to correct. Yeah, I don't agree with some of these. Some of them, like this one, I think are just a segmentation mistake. This one definitely probably belongs in no GFP, but it's right next to something bright, so that could also be a segmentation mistake. But that's definitely no GFP. That's definitely no GFP. These, I would say, have some nuclear GFP. I'm going to go ahead and put them in that bin. Because so many of these were bright, it started to think that some of the dim ones maybe shouldn't count as having GFP in the nucleus. So again, this is where knowing your system and knowing the biology really help. Okay. If I go ahead and I train again, it's getting there. It's getting better. It's still not really sure what's going on with no GFP. But this evaluation also isn't something that I spend a lot of time worrying about. And that's because all this is telling me is that for the 41, 52 images down here, how good of a job CellProfiler-Analyst is doing. And that's important information. You want it to be doing kind of well. But actually what's more important is whether CellProfiler-Analyst has actually really learned the idea of what is nuclear GFP and what is cytoplasmic. So up here is a button I haven't shown you before, which is this random button. Now that I've actually trained my classes a little bit, I can go ahead and decide if I want nuclear GFP, cytoplasmic GFP, or no GFP, and tell it to give me some of those. So these are 40 things that it thinks belong in the nuclear GFP bin. And I have to say, I agree with all of those. I would classify each one of these as nuclear GFP. Some of them, the GFP is not that bright, but I would still say that there's some GFP there. Now what about the no GFP? Because that was the class that seemed to have a little bit of trouble learning. This one is cytoplasmic GFP. This isn't no GFP. And I would say that one is nuclear GFP. It's dim, but it's there. Same with this one. But on the whole, that one too. On the whole, though, that wasn't a bad job. Of the 40 that it gave me, I agreed with 36. And I can now train again and say, now show me what you think no GFP cells are. Can't quite tell for that cell what it should be, but I don't disagree so much that I don't want to include it at all. That one I think is cytoplasmic, but again, now I agree with 39 out of 40. I'm going to go ahead and save my training set again, just because it's been a few minutes since I did that. Just good practice to save. And finally, let's look at the cytoplasmic GFP. This one's kind of got it everywhere. It's so bright I can't really tell what's going on. But it definitely has some GFP in the cytoplasm. So I think I'm okay going ahead and calling it cytoplasmic GFP. Yeah, I agree with everything it did there. So I think at this point, I'm pretty happy with how my classifier is performing. I'm going to go ahead and I'm going to hit score all. And so now, for every single cell in my experiment, it's going to tell me if it thinks it's nuclear GFP, cytoplasmic GFP, or no GFP. Cool. So now I have a table that's been made that shows me for each image how many cells of each kind it has. The number of no GFP is pretty static throughout that, which makes sense since we don't theoretically think that our drug should affect whether cells have some GFP or no GFP. And typically, a lot of these have much higher either cytoplasmic or nuclear. This looks to me like it probably did a pretty good job. I'm going to go ahead and save this table in my database. I'm going to just go ahead and call it hit table. And I want to store it permanently inside my database so that I could always go back to it. And that's it. I've done machine learning now. So I still haven't answered my question though. Ultimately, my question is, what is the IC50 of this drug? how does this drug, what dose of this drug do I need to cause translocation? Let's go ahead and remind ourselves of our dosage over here. So here's our low dose wells, our high dose wells, and our dose response curve. And now with that hit table that I just made, I can open that up in my plate viewer too. I can say that it's a per image table. It's not a new set of images, and the hit table image number is the same thing as image number in this table too. So now I can say, what is the enrichment for nuclear GFP, which is our phenomenon that we think the drug is inducing in each of these wells? And we see here that in the places where dose is high, this is high, and that it seems to go up as we go across our dose curve. This is great because it means that we've probably trained a classifier that tells us where wortmannin is active relative to dose. The last thing I'll do is, again, we just actually want to calculate that number now, is I've looked at these two plots side by side in the plate viewer, but what if I want to actually directly graph them one against the other? So on the x-axis, I'm going to graph my dose of drug, and because it's done in fold change, I'm going to actually make graph it on the log scale. And on the y-axis, I'm going to graph p-enriched nuclear. And you see I have a pretty nice dose response curve here saying that this is about the dose that shows 50% translocation, and that I could go to this dose and still get almost as much translocation as my positive control that's 150-fold, but at 2 to the 2 dose as opposed to 2 to the 7 dose. So we can go down about 30-fold with our dosage and still get about the same nuclear translocation effect. So that's it. In about 30 minutes, we've gone through and trained a machine learning classifier based on measurements we created in CellProfiler to actually answer the question that we had of what dose of drug is sufficient to cause translocation of this GFP transcription factor, and can we do it at a much safer dose than this positive control dose. There's a lot of other things you can do with CellProfiler-Analyst. You're not limited to just looking at individual cells. You can look at organelles inside your cells. You can look at C. elegans worms, for example. You can even actually use this for whole images. When we do quality control for screens in images in CellProfiler, we actually use CellProfiler-Analyst to create a classifier just like this to sort positive high-quality images from negative low-quality images. And if you look at our tutorials page, we actually have a tutorial on doing that too, that you can download the images, download the pipeline, and download a PDF exercise that tells you exactly how to go about doing that in CellProfiler-Analyst. If you have more questions specifically about how to use CellProfiler-Analyst on your data, you can go to the image.sc forum, which is where you can ask questions not just about CellProfiler-Analyst, but about a whole list of tools. And really, even if you don't know which tool to use, you can post, hey, here's my picture, here's what I want to know, what is the best tool to use it. You can keep an eye out for tutorials like this, or even job opportunities or conferences. And I want to thank the Center for Open Bioimage Analysis, which is a new center between my boss and Kevin Lisieri at the University of Wisconsin-Madison that's going to be working to do more algorithm development and community engagement to help bring developments in image analysis to the broader bioimage analysis community, because there are a lot of people who have good images and want to